In the previous video lecture, we talked in brief about the historical perspectives. We discussed why, for a long time, scientists believed that proteins are the genetic material. We also discussed certain indirect evidences that were put forward to prove that it is the DNA which is acting as the genetic material and not the proteins. Those indirect evidences suggested that DNA is genetic material, but they didn't prove it. To prove that it is not the proteins which are acting as the genetic material, it is the DNA which is acting as the genetic material. Certain experimental evidences were put forward. The first experimental evidence which proved that it is the DNA which is acting as the genetic material and not the proteins was given in 1944 by experiments that were conducted by Avery, MacLeod, and McCarthy. The experiments of Avery, MacLeod, and McCarthy were a continuation of the Griffiths experiment. So in this video, we will be discussing both the Griffiths experiment and the experiment that was conducted by Avery, MacLeod, and McCarthy, which is an extension or the continuation of the Griffiths experiment. So DNA is a genetic material, the first direct evidence. So now we are talking about the experimental evidences. The first direct evidence which showed that genetic material is DNA and not protein or RNA was published in 1944 by Ostwald Avery. Now, Ostwald Avery was a physician and a medical researcher. Colin MacLeod, who was a geneticist, and Macklin McCarthy, who again was a geneticist, proved for the first time that DNA and not proteins are the genetic material. So they demonstrated that the component of the cell responsible for the phenomena of transformation in the bacterium Diplococcus pneumoniae is DNA. So the phenomena of transformation was put forward by the experiment that was conducted by Frederick Griffith in the year 1928. So Frederick Griffith put forward the phenomena of transformation, which now we know that transformation is the uptake of naked DNA from the surrounding into the bacterial cell with the incorporation of that DNA into the genetic material of the cell. So that phenomena of transformation, which is a parasexual method, was first discovered by Frederick Griffith in 1928 when he was working on a bacterium known as Diplococcus pneumoniae, which is known for causing pneumonia. Now, the virulence of this bacterium was known to be dependent on a surrounding polysaccharide capsule that protects it from the defense system of the body. The pneumococci, like all other living organisms, they exhibit genetic variability, which can be exhibited with a different phenotype. So there are different phenotypes, and the phenotypic characteristic which was important in the Griffiths experiment was whether a polysaccharide capsule is present or absent. Now, this polysaccharide capsule is known to protect the bacterium from the defense system of the body, and therefore the virulence of the bacterium is dependent on whether a capsule is present or absent. Further, by serological typing, medical researchers sort bacteria into different strains or different types depending on which antigens are present on the surface of the bacterium. So Diplococcus pneumoniae also on this basis is further categorized into type 1, type 2, type 3, and so on. Now, what are these smooth and rough strains? Smooth strain is that which will be encapsulated, which will have a polysaccharide capsule surrounding, and rough strain, which will be the one which does not have a capsule, it is non-encapsulated. So when grown in an appropriate media in petri dishes, pneumococci with a capsule form large smooth colonies and therefore is designated as type S. So this is a type of colony which you can see when this type is grown on the media. Such encapsulated pneumococci, because they have a capsule present, they are protected from the defense system of the body and therefore they are pathogenic to the mammals or they are virulent. They can cause the disease. The other type is non-pathogenic, non-virulent, and because it does not have a polysaccharide capsule, when you grow it on the medium, you get smooth, rough surfaced colonies which are designated as type R. So this is the type of colonies which we can see when we plate it on a media. So these bacterium, they are rough, they do not have a capsule, therefore they are non-virulent, they are not able to cause pneumonia. Smooth are encapsulated, they are protected from the defense system of the body and are capable of causing pneumonia. So Griffith performed a very simple experiment. In the first step, he took the non-virulent type, that was type R, injected it into the experimental animal, and as expected, the animal survived and no bacteria were recovered from the live animal. In the second experiment, virulent type S was taken, injected into the experimental animal, and as expected, because it was a virulent form, it was able to cause the disease. Mice died and virulent type S was recovered from the dead mice. In the third step, he heat killed type S by heating it around 65 degrees Celsius. He heat killed it, injected it again into mice, and the mice now did not die. The mice was alive. Because when you heat kill it, you're removing the outer coating, that is the protection which was protecting it from the defense system of the body. And now it is not protected from the defense system of the body, and therefore it is not able to cause the disease. The mice was alive. Now, in the final step, what he did was he took two non-virulent strains. One was type R, which was non-virulent, and other was heat kill type S, which is virulent when it is live, but when you heat kill it, it is non-virulent. So two non-virulent was mixed together and he expected that the mice should survive. But to his surprise, what he found was that the mice died, and from the dead mice, virulent type S could be recovered. So Griffith's unexpected discovery was that if he injected heat-killed type 3S pneumococci, which was non-virulent when heat-killed, but it was virulent when alive, plus the live type 2R pneumococci, which is non-virulent, when he mixed two non-virulent ones into mice, many of the mice died. But when mice were injected with heat-killed type 3S pneumococci alone, none of the mice died. So Griffith concluded that there is some component present in the heat-killed type 3S cells that is converting the live type 2R cells into type 3S cells. This principle was stated as the transforming principle by Griffith. Subsequent experiments showed that the phenomena described by Griffith is transformation, which is the uptake of DNA from one bacterial strain to another and incorporation of that DNA into the genetic material of the host. In 1944, Avery, MacLeod, and McCarthy continued the experiment that was conducted by Frederick Griffith, because Frederick Griffith could not conclude what is the transforming principle. That transforming principle can be any of the macromolecule which is present in the cell. It can be DNA, it can be RNA, it can be protein, or it can be anything else. What that transforming principle is was finally proved to be DNA by Avery, MacLeod, and McCarthy. So the experiment that was conducted by Griffith was taken forward by Avery, MacLeod, and McCarthy. Avery, what he did was he purified the different components from the heat killed bacteria using the saline soluble components. He precipitated protein using chloroform. Polysaccharide capsule was hydrolyzed with enzymes, and the active portion was precipitated with alcohol fractionation. MacLeod purified the solution of the transforming principle and performed chemical analysis of the active portion. This chemical analysis of the active portion was carried forward by McCarthy and was completed by McCarthy in the 1940s. Then in 1944, Avley, MacLeod, and McCarthy published the results of their extensive and laborious experiments in which they had separated the chemical components, the active portions, they had analyzed them chemically, and finally concluded after chemical analysis that the active portion, which was transforming one bacterial strain into another, was none other than DNA. So the highly purified DNA from type 3S were treated with different enzymes. Different enzymatic treatment was given to find out what is the transforming principle. So this was an experiment that was done by Griffith. He took the heat-killed S component, live R, mixed them together, injected into mice, gave it time for incubation. The mice died and live S were detected. What Avery, MacLeod, and McCarthy did was they purified the different active components, chemically analyzed the components, and concluded that the component which is responsible for transformation is DNA. So they took the live R forms, mixed them with heat-killed type S components, and then treated it with protease. Proteases are enzymes that will destroy the proteins which are present. Injected this mixture into mice, and what happened was that transformation still took place, that is, the mice died. So if proteins were the transforming principle and you're degrading proteins, now they should not be able to transform the type 2 into type 3S. And therefore, transformation should not have taken place. So this proved that it is not the proteins which are transforming, it is something else which is transforming. In the second set, they took RNases. RNases are enzymes that destroy RNA, again introduced into the type R cells, and transformation still took place. Type R cells were transformed into type S when the mixture was introduced into the experimental animal. By ultracentrifugation, fats were removed, introduced again into mice, transformation still took place. So no protein, no RNA, no fat was a transforming principle. Now DNases at that time were not present in the pure form. DNases, a crude preparation was obtained from number of animal sources, and this enzyme could break down DNA. So DNA was broken down by adding the crude preparation of DNases. Once this mixture was introduced into mice, there was no transformation because now the transforming principle or the DNA has been destroyed. Therefore, no transformation took place, and that concluded that it is not protein but it is the DNA which is acting as the genetic material. And when we destroy DNA, transformation does not take place, and the R cells will remain R, they will not get transformed into S. This was a first experiment that proved that DNA and not proteins are the genetic material. In the next video, we will be taking up the second experiment which confirmed that it is DNA which is acting as a genetic material and not the proteins. Thank you.